I want to talk a little bit about clinical application and disease focus. So as EV-based therapeutics move closer to the clinic broadly, understanding where they can make the biggest impact in cell and gene therapy is pretty key, obviously. So how do exosomes, so let's kind of move back a little bit. How do exosomes offer a unique therapeutic modality in these contexts compared to traditional small molecules, or biologics?
Yeah, so that's a great question. I think some of it I've touched on in terms of the fact that their residence time and delivery is one of the residence time and delivery is one of the hardest things to do with drug delivery, right, or drug application, unless it's you're taking a pill and it's just going systemic. So the fact that they very quickly fuse with local cells and dump their cargo in there, hopefully that's what you want, because that will happen, is very advantageous, and you won't get that with, say, a small molecule or and biologics is a big field, right? So that's a big bucket. So I think some other biologics you will get that, and I think there's room for those as well. Things like stem cell treatment, but I've already alluded to the fact that the stem cells don't really stay around that long. I worked at Genzyme for about 13 years, and we had done a lot of studies looking at how long do stem cells stay in an area. So if you put them in a pumping heart, this might sound really obvious, put them in a pumping heart, they last 6 to 30 seconds. They're very quickly gone if you put them in a myocardium. If you put them in a joint, it depends on what the carrier is. If you put them in something thick like a thick hyaluronic acid, they'll be there 24 to 48 hours. If you don't, they'll be gone within 4 to 6. So, and we did those studies kind of over and over in different types of models. And so we do know that we didn't do every exhaustive, tissue type, but we do know that the cells don't stick around long. So I think that fusion mechanism and being able to deliver the value of the cells without having to have the cells is one unique and helpful attribute. I think to compare to something like a small molecule, it's just a very different approach. A small molecule has a target, right? The EVs don't have a target specifically, right? You could say, that we know there is collagen 7 mRNA and protein in there. So when we're looking at a col7 deficiency, sure, they can help develop, help fix that. That's not the main mechanism though, because we know that even without that, we can still have a pretty dramatic effect on the wounds, at least in our animal models. We have, we also know that, like I said, there's just a milieu of other things that are in those. So the target is really the overall regenerative machinery, right? And it's a little hard to figure out, what does that mean? It sounds like hand waving. It sounds crazy. It's a lot cleaner to say, well, we have a small molecule that targets this receptor, and it has this affinity, and it has this, clearance, and that has an excellent place in the world. But it's just a different approach to what we're doing.
Sure. I want to move on to mechanisms and platform differentiation. Now, the precise mechanisms driving therapeutic effects, are still obviously being uncovered. But what do we know today about their mechanism of action, and what still needs to be clarified?
Sure. One of the things I realized that I haven't really talked about much, and I guess this is a good place to kind of bring it in, although maybe I should have talked about it a bit sooner, is our approach is to use native unmodified EVs. There's another approach, and that is to actually engineer the EVs to carry something. So for instance, I'll keep using the col7 example because it's an easy one. If you wanted to actually put col7 in and lots of it and deliver that, I presume you could. We have not tried. We only use the natives. But there are many companies who are working on engineering specific targets into specific ligands, et cetera, into their EVs. And it's hard. It does sometimes break the EVs and make them so they don't, or the exosomes, and make it so they don't work or don't function, or they're not stable. But not always. There's definitely companies that have generated some impressive results with trying to engineer these. So there is that way, and then the mechanism becomes a lot more clear, right? So then you're using the EV as just a delivery to fuse with the cell and dump your cargo in there that you've engineered. With what we're doing, it's a little more nebulous. It's more the massive repertoire of information that the mesenchymal stem cells have that we think really do help in a lot of indications. Not really entirely sure, other than they modulate the immune system, they drive reparative processes, and that's what they're for, right? Mesenchymal stem cells and other stem cells, that's what they're for, right? They're for building, creating tissues prenatally, creating tissues and limbs and things, and then postnatally for repairing said things and homing to those areas and creating that. So our approach is to deliver that mix, and we don't know what's good about that mix, but it's something, and that helps to kickstart and signal the regenerative healing, as opposed to just replacing something.
Sure, thank you. And perhaps this builds on it a little bit. What are the advantages and potential limitations of MSC-derived exosomes from a therapeutic consistency and then scalability standpoint?
Yeah, we touched on this a little bit later, and I see the results of the poll up there, and I wholeheartedly agree with our audience that is the most significant challenge, is scaling up manufacturing and ensuring batch consistency. Consistency we found is a little less hard than the scale up, and just making sure that we're still finding our way here, right? And we have a panel of release and stability assays that characterization that, at this stage, they're stage appropriate, and the FDA has accepted those, and we're going forward with those. But, as we move into, we're starting looking forward to pivotal stage, and that's a much higher bar. And so, how do we get there with pivotal ready material? And we're doing a lot of work in the background to get there, as are other groups, and we're working with some teams that are not necessarily well known in the world for EV manufacturing, but they're known for similar types of things, and they're helping us develop that. So that's still a tough nut to crack. The scale up, I don't think it's impossible. I think it's definitely doable, but it does, as I mentioned before, it does take the systemic application is going to have to be looked at very carefully, because if you need more than one application, and don't know if you do or don't, that's still unknown, that's going to be something you have to contend with. How many patients can you treat, and what indications would you go for if you know that you might be limited by how much you can create? And that's still something we're working on.